What if aging is not a broken machine, but a corrupted operating system? Imagine your phone slowing down, overheating, crashing, not because the hardware is destroyed, but because the software accumulated errors over time. Now imagine you could restore the original code without replacing the device. That idea sounds like science fiction, but in biology, it's starting to look like an engineering problem. In 2023, researchers linked to Harvard showed something remarkable in mice. When they deliberately disrupted the epigenetics program, the cell's regulatory software, the animals aged faster. And when they partially restored that program, the mice showed signs of rejuvenation. So the real question is, are they learning how to reset biological age? Or are they just watching clever experiments that won't survive contact with human biology? And if aging is so complex, where do we even begin? Welcome to our longevity series. I'm Elena. In Gen Explain, we look at longevity not as a miracle pill problem, but as a systems biology problem. Aging is not driven by one gene, one pathway, or one molecule. It's driven by regulatory programs and by the key regulators that control them. For many years, this has been our focus, reconstructing gene regulatory networks from multi-omics data to understand what truly drives complex biological processes. This is episode 1. Can science turn back the clock on aging? Here is the first key point. Aging is not caused by a single factor. It's a multi-layer, interconnected drift of biological systems. Scientists often describe aging using the hallmarks of aging. For example, DNA damage accumulating like tiny typos in critical instructions, telomeres shortening, worm protective caps at chromosome ends, epigenetic regulation shifting, so the same DNA is read differently over time, mitochondria producing less energy and more cellular stress, chronic inflammation, keeping the body in a constant low-level alarm state. This is why statements like "this gene controls longevity" are almost always oversimplifications. Aging happens when many systems lose coordination at the same time. Instead of treating age-related diseases one by one, researchers are now targeting aging itself, the shared root. Here are 3 directions that truly matter today. First, clearing senescent cells, Senolytics. As we age, some damaged cells enter senescence. They stop dividing, but they don't disappear. Instead, they secrete inflammatory signals that damage surrounding tissue, like toxic neighbors that slowly ruin an entire street. In mouse studies, removing senescent cells improve health and extended lifespan. That's why Senolytics became a major focus. The challenge is precision. Remove the harmful cells without damaging healthy ones. Second, epigenetics reprogramming, the software reset. DNA is the hardware. Epigenetics is the software layer, deciding which genes are active when and how strongly. Partial epigenetics reprogramming aims to restore youthful regulation without erasing cell identity. This is the deep implication of Harvard-style experiments. Cells may retain a backup copy of youthful regulatory instructions. Importantly, this isn't limited to genetic engineering. For example, resveratrol, a plant polyphenol, has been shown to influence chromatin regulation and TF activity via SIRT and FOXO pathways, acting as a mild, indirect epigenetics modulator rather than a full reprogramming trigger. Quick note: if you like this evidence-based approach to longevity science, subscribe. This is a short series with several and you risk cancer. And if you want more details, our blog article is linked in the description. Let's continue. Third one, metabolic and dietary signaling. Caloric restriction extends lifespan in many organisms by activating survival programs. Increased autophagy, the cell's internal cleanup system. So, researchers study caloric restriction mimetics, enhanced DNA that trigger similar regulatory programs. These include metformin, rapamycin, NAD, pathway modulation. But long-term caloric restriction is difficult dietary in real life that reshape transcriptional and epigenetic states. Again, the theme is not magic molecules, but regulatory controls. Longevity is not about isolated genes. It's about regulatory blueprints. Control circuits deciding which programs run under stress over time and across tissues. That's why transcription factors are so important. A well-known example is FOXO3, repeatedly associated with exceptional human longevity. But FOXO3 is not in mice. Altering these signals affects muscles, brain, and immune conductor, coordinating stress resistance, metabolism, blood therapy, DNA repair, autophagy, and immune balance. Its power comes from network position, not from acting alone. Let's be honest. Most dramatic rejuvenation results still come from model organisms. The real bottleneck. Interventions that affect gene regulation demand extreme caution, long-term safety, reproducibility, and systems-level validation are essential. Optimism is good, hype is dangerous. Today, they build the map. Aging is a systems-level process. Multiple scientific paths are converging and partial rejuvenation. The key lies in understanding regulatory networks. In episode 2, we'll focus on epigenetics regulation in magic detail switch and demonstrate how genome-wide DNA methylation data can be analyzed to identify master regulators of aging using genome enhancer. If you're enjoying this episode, like, subscribe, and comment. Which direction do you find most promising? See you in the next episode. Their science starts to be more complex, a little like science fictions and lot like engineering.